Hello. So this is my elevator speech about gene editing and the ethics involved in it. First, let me just start by explaining, based on that New York Times article that we read, they were using CRISPR-Cas9, and that's a genome editing technology that cuts DNA at the target site of a gene of interest, and the short DNA sequence unique to the gene of interest is used to design and guide the RNA. And then that guides the Cas9 protein to the location where it will cleave the gene and creating a double-stranded break in the DNA molecule. So when the DNA repair mechanism attempts to rejoin the strand, it either creates indels, insertions, or deletions of nucleotide sequences within the gene, which can disrupt protein production. So that's basically how CRISPR is being used, to knock out or knock down, upregulate genes, and can also be used to repair damaged genes. An obstacle in medical application is that right now it's just being used in vivo, so it's just animal research, one cell stage. And the studies in vitro, it's the monocultures outside of the organism. So they involving human cell lines that show promising results in efficiency repairing sickle cell mutations is what the article from Huang showed. Unfortunately, research has yet to reveal a way to entirely repair faulty genes in the germline once the cells have differentiated in an embryo and past the one cell stage. One study revealed partial restoration of dystrophin protein production and muscular development in young mutated mice by using viral transfection of CRISPR constructs to modify somatic cells. And that I found that in this article by Nelson et al. The study was a muscle cross sections of mutant mice that underwent CRISPR therapy, resembled wild type control mice, and the desired therapeutic levels were achieved. However, does not erase that a heritable deleterious mutation still exists within the germline on some level and can be passed to the next generation. So the ethics, involved with all of this has to do with creating potential other problems once you start cutting into genes and cutting into DNA sequences to remove things. Life has a way of creating more life, and the potential there is to create new mutations, and mutations that our world has never seen before. And what do we do then? Do we just keep going, And so genomes also vary between organisms greatly, and the results of a CRISPR target used in mice or any other model organism will not necessarily translate to humans. So once experimentation has been performed exhaustively in model organisms and a solid understanding has come, then we are ready to move to humans, which will require lifting of moratorium on human germline modifications. Though researchers have rapidly progressed with the field of gene editing by applying CRISPR to an array of non-model and model organisms, studies have revealed that genomes control a vastly greater and complex system than ever realized. So we're far from moving to humans as for now, but hope is still there, and technology. I mean, we're still discovering DNA, like what it is. New things keep coming up. They just recently created a chemical that is injected into a dying cell, and the cell rejuvenates itself. So things are moving, things are shaking, but again, human research, I think, out loud human research. I'm sure there's some laboratory somewhere in some dark basement going on, but as far as like up and up, I think we're far off. Anyways, have a great semester, and thank you.